Hi there, let's talk about fermentation. Fermentation only involves the glycolytic pathway and it only produces a net gain of about 2 ATP. So it's very minimal amounts of energy. So this is that glycolytic pathway. I told you that some organisms only use glycolysis as their mechanism of energy production. And so this is that fermentative pathway that we're talking about. In fermentation, the final electron acceptor is going to be pyruvate, which is an organic molecule. And the function of fermentation is to regenerate NAD from NADH so that we can continue the glycolytic pathway. Hello, there we go. And so here you can see that glycolytic pathway and then what happens afterwards. So there are two major types of fermentation. There's homolactic fermentation, where the end product is lactic acid. And so this is what humans, other animals use, and some bacteria use. They produce lactic acid as their end product. And so we take glucose, we break it down to pyruvate, and then the pyruvate is reduced to lactate in the process of fermentation. But then we also have another mechanism called heterolactic fermentation, where we produce a mixture of lactic acid and ethanol or acetic acid, okay? And so here is that heterolactic fermentation. So in this process, we're going to take pyruvic acid and we remove a carbon. So we have a decarboxylation process, and we produce acetaldehyde, and then we accept electrons from the NADH. So we add those electrons and hydrogens, we reduce the acetaldehyde, and that produces ethanol. And again, we have oxidized NADH back to NAD so that we can continue that glycolytic pathway. Fermentation is used in multiple processes for food making. So we produce alcoholic beverages, we produce breads, we produce yogurts, we produce cheeses, we produce biofuels in a fermentation pathway. And so different organisms are going to produce different end products of fermentation. And so here we have different types of organisms and what they can produce. So aspergillus, which is a fungus, can produce lactic acid, and with lactic acid we can produce things like soy sauce. Lactobacillus uses ferments to lactic acid, and with lactobacillus we can produce cheeses and yogurts. Saccharomyces, another fungus, this is a form called yeast. We can ferment producing ethanol and carbon dioxide, which forms beer, ethanol, wine, carbon dioxide, bread. But just like all living things, we can break down other molecules, or microbes can break down other molecules to produce energy. And so they can break down lipids or proteins. They have enzymes like lipases or phospholipases, which can break down triglycerides or phospholipids into smaller subunits. They also can utilize beta oxidation, similar to how we break down fatty acids, which then can produce a lot of reduced NADH and FADH in the citric acid cycle. They also have proteases that they can use to break down proteins into smaller peptides, and then these peptides can then be broken down into their specific amino acids, and these can then allow those organisms to use those amino acids. And the different types of proteases that they have can be used to help differentiate different types of bacteria as well. So now we're going to get into another pathway. So we're going to talk about, instead of breaking down glucose or breaking down fats or proteins, we're now going to look at building up carbohydrates by the utilization of sunlight energy. And so this is photosynthesis. So heterotrophic organisms, which are like me and you, as well as E. coli and Enterobacter, we are organisms that depend on carbohydrates that are produced by photosynthetic organisms, such as plants and algae and cyanobacteria. So in different photosynthetic organisms, different photosynthetic pigments are going to be found. So when we talk about photosynthesis, which I told you, different organisms that use it, we're looking at, we're taking in inorganic carbon in the form of carbon dioxide. We're going to use some type of molecule to, this is going to be called a reducing agent. It's going to get oxidized. The one that we typically think of is water, H2O, but this could be in an anaerobic organism, it could be H2S, okay? So that X just means some other molecule. We take in sunlight energy and we convert the carbon dioxide into glucose, so we reduce it to glucose, and we're going to oxidize this to something else. So if this is water, we oxidize it to oxygen. If this is hydrogen sulfide, then we oxidize it to sulfur. Okay? And then we produce energy as a byproduct. Or, I'm sorry, and so we use energy as an input. We're producing some energy to for the process, but we use energy as an input in this case. So the different types of pigments are going to be found in, organized together in photosystems. So in eukaryotes, these photosystems are found within the chloroplast. But in prokaryotic organisms, they're going to be found within the cytoplasmic membrane. So there are two major types of reactions that we're going to have. We're going to have light-dependent and light-independent reactions. Light-dependent reactions are going to take sunlight energy and harness that energy to produce a lot of ATP and reduced NADPH. And then we're going to use those molecules in the light-independent reactions. So light-dependent reactions, there are two types. There's a non-cyclic pathway and a cyclic pathway. Non-cyclic photophosphorylation is used in oxidation or oxygenic photosynthesis, whereas cyclic photophosphorylation is used in multiple bacteria where they're going to need a lot more ATP than they need NADPH. And so a lot of those anaerobic organisms, they're going to use the cyclic photophosphorylation. So here, this is from the Openstax General Biology textbook. This shows over here the cyclic pathway and over here a non-cyclic pathway. So we start with this photosystem 2. Water drops off hydrogens and electrons. The hydrogens then are going to move into an electron transport chain. The electrons are going to move into the photosystem, and they're going to move up in energy. As sunlight energy comes in, the electrons get more and more excited. This is going to cause them to jump into an electron transport chain. So this electron transport chain then is going to produce ATP as the hydrogens move across a membrane. They're going to produce ATP. So these hydrogens, after they go through the membrane, they're going to then attach to the NADP, which is waiting, and they're going to become NADPH. The electrons are going to move into photosystem 1. So they move from this system to this system, and they're going to move up in energy levels as more sunlight energy comes in. This is going to cause the electrons to get higher excitable, and then they're going to jump, and they're going to also attach to the NADPH that is now there. And then the NADPH, which is now holding a hydrogen and an electron, is then going to move into the light-independent reactions. In the cyclic pathway, all we're producing is energy. So here we have photosystem 1, and the electron, so there's no movement. The electrons that we have don't come from, or are not being replaced, they're replacing themselves. So photosystem 1, the electron gets excited as sunlight energy comes in. This causes the electron to jump and move into an electron transport chain. Hydrogen ions are pumped across the membrane. They move across the ATP synthase molecule. And when that happens, ATP is produced, and then the electrons, lower in energy, move back to that photosystem 1. So that's a cyclic pathway. Oxygenic photosynthesis, or in oxygenic photosynthesis, water serves as the electron donor, and oxygen is going to be formed as a byproduct. In anoxygenic photosynthesis, some other molecule like hydrogen sulfide or thiosulfate are going to be used as electron donors. And so this means that oxygen is not formed. And then in the light-independent reactions, the cell is going to fix carbon dioxide and produce glucose as an end product. So here's that Calvin cycle, or the light-independent reaction, okay? So this is, again, from a different textbook, but it's still showing what's going on. So we have carbon dioxide coming in. It's going to be fixed to this 5-carbon molecule called rubisco. And that forms a 6-carbon molecule that is unstable. The 6-carbon molecule immediately is going to break down into 2 3-carbon molecules called 3-phosphoglycerate. The 3-phosphoglycerate are then going to be reduced into glyceraldehyde 3-phosphate. And if we take 2 glyceraldehyde 3-phosphate and put them together, we produce 1 glucose. But you're going to have this spin a total of 6 times to produce 1 glucose molecule. Because down here, the end product is going to be these 3-glyceraldehyde 3-phosphate. And if it spins 6 times, for each spin, we produce 2 glyceraldehyde 3-phosphates. So the majority of these glyceraldehyde 3-phosphates have to move into producing or reproducing the regeneration of RUBP, because remember, this is a cycle, so we have to reproduce the original product. And so very little carbon is actually going to be used to produce the glucose. So for 6 turns, this is saying 3 turns, we get 3 carbon sugar. For 6 turns, we'll get 1 6-carbon glucose, and the other 5 the other molecules are going to be used to produce the RUBP. There we go. All right. Actually, I'm going to stop here because we have very little left to go, but this has been a 15-minute video, so I will start the next video shortly. Bye.